I'm going to show some slides about this. I'm not going to tell you all the deep engineering details of this because I think that's not really needed here. But gee, I wish I could make this go into a big form without going through the whole
Can you right-click on the slide?
Oh, right-click.
So technically, this patient did their finger sticking correctly.
Really?
Yeah. They should actually be a little bit closer to the tip to prevent minor nerve damage.
To the finger?
Yeah.
The spot, the little blood spot?
Yeah, it's a little bit too close to the center of the fingerprint.
See that?
They should be a little higher up here, actually, or actually close to the sides, to be honest.
There's a button, it's either like F4 or
F4?
F5. Try it one of those.
F5.
F5. See what that does.
F5 would just make it darker and lighter.
Go to the home page.
I apologize for this. My other system, you can start your slideshow in the middle of the thing, you know. It's,
So there's a slideshow on top.
slideshow, yeah, but it'll go
There's a
right back to
slideshow
the
button.
first
You
one.
go to the first line.
I think you can probably,
It starts back all,
the other one you can stay from this side on, but anyway, okay.
we gotta get better software for this. I don't wanna go through a,
Office 2007 is supposed to be new and improved.
yeah.
And all it does is frustrate people.
Okay, I'm skipping lots of this engineering stuff because you're gonna hear about engineering like crazy later. And this class is not so focused on all that. Let's see, I missed the whole one. I wanted to show you. I apologize for this. But, you know, then I got thinking the other day, gee, if I have all these slides disorganized, then there's a big temptation to just go show slides one after another, and somehow that's not very spontaneous, and, you know, it's not the right way to go. So there's that guy, here's the artificial paint, we were talking about that before. Okay, oh, let's see. This one I want to show you. There's a better version of it later, though. Sorry. So,
do you all know what body mass index is?
Mm-hmm.
Does it make any sense to you?
It's just an equation, right?
Yeah, the equation makes sense, but the use of it is, I think, overused.
it's the only thing the docs have, though, as far as, you know, that's their main tool nowadays, right?
Well, I don't. Anyway, when I talk later, I'll tell you the history of the body mass index.
I'd like to hear that. Okay, so we had the finger stick. So here's what I'm trying to make, okay? Instead of making the full artificial pancreas, other people are making insulin pumps and have done that for a while, and you know, that's fine. And there are people that are controller oriented and want to use the signal from some kind of a sensor to control those pumps. That's a world. This is the world we've been in. And this picture is historic. This is made in 1972 or something like that, you know. This is one of my mentors back at Boston. He said, okay, this is my dream. I want to have one of these things someday, you know. So it's got a glucose sensor, it's got a power supply, it's got a transmitter, and then all that's parked under the skin, and then you have an external receiver. Okay, so this is pretty hypothetical in those days. Remember, engineers were still using vacuum tubes in those days, right? So some of this kind of stuff is a dream. And you know, there's been some advances on it. So we got busy with this idea. I'll just tell you the basic physics of it. I made a sensor that uses two enzymes. These enzymes, of course, are biological catalysts, and you mobilize those into a little membrane, and you put them over an oxygen electrode, electrochemical oxygen sensor. And these enzymes catalyze these reactions here. So glucose diffuses. Sorry, this thing's not so effective, but here's the two enzymes here in a little gel. And when glucose diffuses into that gel, and oxygen diffuses in there from the body, then you have that top reaction. Glucose plus oxygen makes gluconic acid and peroxide. And we have a second enzyme, catalase, which takes peroxide and breaks it back down into half oxygen and water. So you have these two enzymes together, the glucose plus half oxygen goes to this product. So those are sitting in there, you have this oxygen sensor, and then you have a second oxygen. So that makes this what we call glucose modulated oxygen dependent current. So there's a current that's produced when oxygen reacts at the oxygen sensor, and it's directly related to glucose. And then you need to know how much oxygen is present in the body to begin with, right? And this is an oxygen sensor without any enzymes on it. And so this makes the oxygen dependent preference current, and you subtract the two and you get a glucose dependent difference current. Okay. So that's the physics of it.
Do you actually need to neutralize the hydrogen peroxide?
Yeah. Well, the way this works is, of course you want to get rid of hydrogen peroxide because that's a very strong toxin, right?
Yeah, what actually used to be used for treating arrhythmias.
Yeah,
it was
well,
pretty effective.
It causes protein denaturation, and
but it's such a small amount.
You can, for me and you it is, but the FDA doesn't look at it
It's
as
a lot.
Okay.
I see.
Okay, yeah. I'll complain about the FDA later. More effectively, put it in bigger prospect. Anyway, so this idea, we started working on this in the 70s, right? And this idea, the idea of, well, if you're going to make a device like that, it has to last long enough that you can justify implanting it. You can't just implant something for a short term and then, it's got to stay in there for a long time. And so I used to write grant proposals, and people pointed out the defects in my thinking here on this. One of which was enzymes are unstable. If you have those enzymes, you're going to do a reaction with them sitting on the bench here. In 2 hours, they will have lost activity. And they'll be totally gone in a day, okay, at body temperature or room temperature. So, I got lots of critiques for saying, oh, this is a great idea, this will be fine, but it won't last for more than a few days at best. And because don't you know that enzymes are unstable? Okay, I guess I didn't know that at the time. And then there was this question from the electrochemist. Well, the classical Clark oxygen electrode that's widely used in clinics, is not stable. Okay, and well, how come it's widely used? Well, because you recalibrate it after every measurement. When you go to the doctor and they're going to test your oxygen level in blood, take a sample, put it in there. The machine measures the oxygen, but then it automatically substitutes a calibrant solution which has defined oxygen between the next sample. Okay. And the Clark oxygen sensor is used in the United States for like 2 billion oxygen sampling events in a year in the hospital, right? It's a fabulous tool, but if you try to run that thing steadily, you find that it just decays and the story's gone. And so, okay, that's another big problem. Got to make an oxygen sensor that's stable. And then, of course, you have, I'll skip some of these other things. You got to make an implant that's biocompatible. It's got to work in, and not too many things are real biocompatible. Put them in the body. Body's not too happy about that, and they try to isolate it some way, and try to get rid of it, and try to defeat your goal. And there are other issues here. These are just kind of incidental things, don't feel like there's going to be any quiz on this or anything. I'm just telling you some of the parts of the developmental story that there's similar things in every medical device issue, Anyway, so we first, years ago, so I want to tell this part of the story that, years ago, we said, okay, let's make a needle sensor, okay? And this was sort of stimulated by industry, Alman's company, and said, okay, we'd like to make this a little needle sensor, a very fine needle, and you insert it under the skin, you tape it in place, or you hold it in place with this fancy stabilizing plastic part here. The needle's underneath here, it doesn't really show. And there would be a connector that either telemeters to something or just a wire that goes to a display tank. Okay? And so we did a lot of work. We made a needle version of the sensor. This is, just to show you what the needle part actually looks like in a patent type picture, right? And
razor blade?
Huh?
On the right, is that a razor blade?
Cross section.
This cross section of the needle.
okay.
And these are there's two sensors on the side of this needle here, basically, and internal structures. So it's just a 2D representation of a 3D object. And we got a patent on it, and the industry got all excited about this. But then, the problem was, I remember one key meeting going to the industry guys with Al Mann there, and we were talking and saying, this thing doesn't work well. Because you can put this in, and it creates a wound, and it takes a couple days for the wound to actually stabilize. But the FDA won't let you have a percutaneous device for more than 72 hours. Right, because it can get bacteria, you can get bacteria growing. You can't have something sticking out of your skin for more than 72 hours, right? That was the rule that the FDA imposed just because bacterial problems so obvious. So we don't want that. So you had to change this thing out every 3 days. And industry got all excited about that because that's a disposable. This hit the magic word that, okay, that's fine with us. We want something you have to buy over every 3 days, buy another one, right? Good. And industry was so excited about making this because it's, the smaller you make it, the better, right? All this kind of thing makes sense. Except that when you put it in place, the signal decays dramatically during the wound healing process. And so you still have to finger stick if you use this device, okay? And the way I was thinking at the time is, well, who's going to be willing to finger stick to recalibrate this and put this thing in and go and then change it out? Who's going to be willing to do all that? So I figured it's a non-product. It can't work. This is my thinking, okay? And obviously it is a product. So what happened? Well, there's one smart guy in industry that said, okay, it doesn't work for measuring glucose. You can't use it as a primary glucose standard, okay? But maybe you can use it to measure glucose trends, okay? Once it's calibrated, whatever level it is, you can see if your glucose is going up and down. You don't know what the absolute number is of glucose, unless you do finger sticking and sit there. But it could be used to follow trends. So they went to the FDA with that labeling plan, right? Said, okay, this sensor it's not a glucose sensor, it's a glucose trend analyzer. Okay? And I thought that was actually pretty smart. Otherwise, you don't have a product, So somebody in industry got much smarter than I was and figure out how to do this. And, well, what happens when your glucose starts going down, if you read the little product insert, it says disregard the sensor and start finger sticking, okay? Because it's not approved for hypoglycemia, it doesn't work in that range. And you have to agree to your finger stick to run with it. So, so I just couldn't imagine anybody be willing to do that. But, there are a lot of people who are willing to do that, okay?
So what does it actually tell you when you become hypoglycemic?
Hyper, yeah, when your glucose is going up, okay, then let's go exercise, or let's maybe adjust our medication or something like that, And that's really what it, but, so the FDA requires that labeling, glucose trend only, right? Patients use it any way they want, okay? Some patients ignore that and they say, I'm going to use this when I sleep at night, it's going to tell me to wake up. It's not approved for that and it's pretty dangerous, but I don't care, I'm going to use it that way. And so, I talked to a large number of patients, and people that use this is the number, this is going down, it never went higher than this, 300,000 patients. And that's a really small fraction of the diabetic population, right? Could exist. So these are early adopters, they're really aggressive about their health care, These athletes that run a marathon, they're diabetics they want to have one of these while they're running, you know, and as opposed to, you know, okay, they got to do finger sticking and all that, but they swear by it. A small group of people just love it because they were in such bad shape without this. Okay, that's the only answer you can give. What life must have been absolutely hell without this, right? They're willing to do this. And of course, you talk to any of them, and they'll show you the site. Gee, I put you know 300 of these in over the last 5 years, and look at this site here. You know, I mean it's bad, right? And that's the first thing they do when they talk to you is they look at my site, and I mean, you know, you're putting this needle in, taking it out, and once it's in, of course, it's not totally immobilized, and so yeah, there was a big worry just recently because there's no instructions on what to do if the needle breaks. Okay, on the package insert, companies didn't put the instructions in there, so people, when the needle breaks off in place, some people try to figure out how to get it out, right? Whatever tool they have handy, right? Making a big mess of everything, and so the company that makes these failed to report these incidents to the FDA, right? So this is a big violation. You know, you report all incidents to the FDA, and this one wasn't planned. Who would ever think this would be an issue? These things would break, little tips would break off and stuff. Every now, not very often, very rare event, okay, but you got to report that stuff, and they didn't do that, and so there was a big flap about that. But you know, I mean, so there are some patients that use this, and the UCs receive royalty income from this, you know, yeah, from that patent. And, but I'm not saying it's a solution, okay? Yeah.
So you developed the patent, or you got the official patent number in 1987, so it took you 20 years to get to market?
Something like that.
Okay.
I'd have to tell you exactly, not exactly 20 years, but something like that.
Well, it's simple math.
Yeah. And so the patent life was almost gone. I think we probably had 2 years of life on them when they actually started selling these things.
Okay. You can't extend the patent. What's the...
Oh, you write new patents that have, you know, they have a patent on this little plastic part, you know, this, okay, and then there's an inserter device that holds it just right, you know, that's right. But they're kind of tied into the original patent, but you know, unless you can come up with new ideas, you cannot write new patents, right? So that's a fact of life. Thank you for bringing that up. You know, the other message that you should definitely have in mind is that medical devices take of order a decade, not years to develop and make, right? And so when you get a patent, you know, it's a race to get the device into the marketplace before the patent dies. If that's your motivation, right? You know, sadly, it's that way, and all medical devices take so much longer. You know, that's one of the things that's so different from the other group is thinking about wireless stuff. You know, you can make a wireless gadget, you know, and you can make it in a couple of months, and you can have it on the marketplace in a year, you know, even faster, right? People buying it, okay, the company's going along just fine. That ain't gonna happen in the medical device area. Okay, I'm sorry to tell you this. And since I'm lecturing on that subject right now, it's why is that important? Well, you've got to raise money to do this somehow, and you've got to have a strategy that doesn't require you to give the money back next year, right? Most venture capitalists are thinking in, you know, they want to turn around a new software, a new computer, or something like that in a year, and they want their money back, plus all the money they made on it in a year. And if you go up to Silicon Valley up here and talk to the VCs on Page Mill Road up there, where they're all located, right? They all think that, not all, but there's a big group up there that thinks, how fast is we going to see new Google in a year, you know, or something like that, right, from where we are now. That's the turnaround time. Fortunately, there's a group of investors that are medically oriented, and they have a longer time scale in mind, okay? It's a big deal to say, well, we're going to take your money, we're going to work on it for 10 years, and so don't call us back for 10 years.
Good one.
Yeah, but that's the facts. You know, but
yeah, sure.
I'd like to find a device that hasn't, 10 years is a record fast time for a device, really, you know?
Sure.
If you look at the history of all these. So that's a big, big item that this class specifically is, you know, that's a big message. Is you got to figure out a business strategy to survive for 10 years, approximately, in order to get something like this done. That's a big challenge. Most companies have lifetimes that are shorter than 10 years, you know. Anyway, so this version here, and this shows, here's a insulin pump, okay, made by somebody else. There's half a dozen companies that make these now, and you can program it. It pumps insulin through a little tube, and there's another little needle sitting there. This is totally limited. You can do about a basal level of insulin, and then about a 10% above that level, and then it just won't pump anything beyond that. So it's not like you're going to control all these big excursions with this thing at all. This isn't possible. But it delivers insulin at a pretty uniform rate during the day, right? And patients who use this have to finger stick like crazy, and you know, when they go to eat, they can adjust. They're going to have a big meal. They can adjust it up a little bit, but not close to optimal control for insulin delivery. Now, I have to emphasize that insulin is a pretty unusual molecule. Excuse me. Because it has such a small margin of safety. Margin of safety in the pharmaceutical world means, you know, there's a range of dosage that you're ideally supposed to hit. And well, what if you hit twice that? Is it dangerous to you? You know, what if you hit 10 times that? Is it dangerous? Because people misjudge their dose all the time, you know, or get the wrong dose for some reason, or when you inject insulin, instead of injecting it into the subcutaneous tissues, maybe it goes directly into the bloodstream. Well, I didn't plan on that, you know, and you might end up getting a much higher dose than you planned. Well, so insulin, it's dangerous because, sorry, it drives your blood glucose down very sharply, like I've shown in some of these slides, right? And if it drives it down below 40, you don't even know what hit you. I mean, you won't have brain functioning, and you won't know how to readjust anything. And, you know, this is the problem. If you take twice as much insulin as you need in a single dose, you could kill yourself. Okay. If you take, compared to penicillin, if you take 1 million units of penicillin, it'll get rid of your bacteria, but it won't do a thing to you. You know, you're going to be just totally happy, right? It's not going to kill you. Well, if you take 10 million units of penicillin, same story. You know, it's just fine. It has an enormous margin of safety, right? And insulin, however, has a very, very small margin of safety. So docs know that. They're not going to just give a patient this and say, okay, crank it up any way you want. You know, this is a very narrow range of insulin.
That's why OG goes to 110%. You know,
Yeah.
very limited.
Yeah. So you might ask yourself, wait a minute, can't you go to the pharmacy and with a prescription buy a bottle of insulin? Yeah. The answer is yeah. And wouldn't that create a big problem for the pharmaceutical industry selling that? You know, what if, you know, this is so dangerous, what if people just use that and inject themselves too much? Well, you can't buy too much insulin. If you try to buy a, you know, they give you a pretty dilute level and you can't really get that much insulin. And second, the insulin is specifically made to be slow acting, right? Zinc insulin. And that means, okay, you give yourself an injection and it's going to have its effect over, you know, 4 or 5 hours or something like that. And drug companies are totally aware of the liability that would be associated with giving people enough insulin to commit suicide. You know, they don't want that kind of thing to happen at all, right? So, but now there's a big issue. If you look at that diabetes plan I mentioned, there's a group of people saying, oh, we need super fast insulins, okay? And there are groups working on fixing that problem, right? So they'll absorb really fast, and so your glucose starts going up, you can cut it right back down by quick absorbing insulin. That's like a big nightmare from a control perspective, because, well, what if you cut it a little too low? No, you know, this is happening too fast, and didn't plan on that. So that's a big concern. Puts more, the reason these companies popped up is because there are glucose monitors that are coming onto the market, and okay, we're not so worried now because we have some way of deciding on the liability of this. If you give yourself too fast an insulin, we can monitor that with your glucose levels, right? And therefore assign blame to this later. But I'm just saying that this pumping strategy here is pretty minimal. Okay, it's not really a close control. This picture here is totally doctored, as you can see. This means an RF wave here controlling this. No, it's not like that. Okay. This is, but anyway, just to tell you that okay, there's you know, a marketplace for this kind of stuff, and it doesn't really solve the bill, and patients still don't like it too much, but nevertheless, it's there. Now, here's a oh, let's see. Can I tell you about this other? We have several versions of sensors we've made over the years. Some are big disasters for reasons you need to know. We made another sensor years ago. I'm sorry to do this, but this is my idea of spontaneous talk here. We made another sensor that came after the one I just showed you, and it looks like this. Okay, this is the same mechanism of sensing I mentioned before. You know, and one of these is it's got a little oxygen electrode here with a membrane glucose oxidase on it, and this one is just the oxygen reference electrode, and then this is an antenna. Okay, and this was built by my students. You know, and this uses a battery that you can buy at Vons, right? And this is a epoxy encapsulation, and we're thinking, okay, this would work for a few days and try it in dogs. Well, it worked for quite a while. The sensor tip was implanted in the superior vena cava, the big blood vessel from above, right? And the telemetry unit then is just put under the tissues, right? So here's a dog, here's the telemetry unit, and then here are wires that are part of the sensor that go down into the tips. The tips are close to the place where the blood flow takes a left turn and goes off to the heart. Okay. And the reason we put it there is because it's in direct contact with the blood, so that would have the fastest response you can imagine. And the vena cava, the superior vena cava, is a pretty privileged site as far as blood clotting goes. Almost anything you put in the bloodstream, if you try to put it on the arterial side, it'll clot in 10 minutes, right? If you put it in most venous sites, it'll clot probably in about a day or so. Okay. If you put it in the vena cava, the blood flow is smooth and slow, and it's not pulsatile, and it kind of washes off anything. And if there's any thrombus that are formed on it, then the blood goes to the lungs. And the lungs act like a big filter, and they're going to digest any of these little floating emboli. So that's the same strategy that the pacemaker industry uses. They put their pacemaker leads in the superior vena cava. They have to have the tip of the lead stuck in the heart wall. Okay, this one's just sort of free floating in the center, right? So it's like the ultimate great way to measure glucose. It's the gold standard of glucose, dynamic glucose. Right? I mean, if you're willing to do all this, you can get the fastest glucose measurement, follow the most dynamics, all that stuff, up any place in the body. And anyway, so we tried this in dogs, and we got great results. Here's the example of the dog. Went for 100 days, several months without recalibration. And at that time, that was absolutely a major milestone, because people were saying, oh, the problem is those enzymes are supposed to die in a few days. You know, this is going on for months. Right? You can recalibrate it later, check the calibration, no change. That's fabulous. So this was passed off by the university to a medical device company here in California, and they made a human version of this. Okay, and here, just before I get to that, here's the dog study. This is time and measurements, and then this is glucose on this side, and the signal from the telemeter device is this black line, and these little triangles are blood glucose for a sample, you know, to try and compare with the telemetry thing. So this is on day 108 of implantation, okay? means the enzyme's got to last that long, and biocompatibility's got to be that long at least for this to happen. We did this in a number of animals, and these are normal dogs, and you have to give them a glucose injection in order to get anything to happen in these dogs, right? So here's 2 identical glucose challenges in a normal dog, okay? And, you know, giving 2 identical challenges in a row does not give identical glucose excursions, okay? So there's some nonlinearity and non-stationarity in the system there.
So the dog can't just feed the dog to get a glucose, you have to.
It takes a long, long time, and it goes much slower, and you know, sometimes it doesn't go very high volume. And it depends, some dogs you can't get that to happen because they're just very healthy, and other dogs, you know, you gotta find one that's, so that's not great experimental protocol, you know, that just doesn't get you very far.
How do you finger sticking problems?
Oh, you can't, you can tell them to put their hand up here, but the best way is you put a catheter in there and draw blood through a catheter.
Okay.
And the catheter can be permanently in place.
Okay.
So anyway, that's the work of my students. This is before implantation and after explantation. The signal doesn't change that much. No. So the company ran with this. Okay, the university licensed the patents we have to the company. They made a human version of this. Okay, same basic idea, everything. And this is what it looked like, right? They started clinical trials, and clinical trials were designed so that it can be 2 years. So each patient is going to have one of these in place for 2 years, and if their blood glucose is 25% or more different from the sensor signal, then they're going to discontinue that patient and take the sensor out. Okay. And so they had a number of patients that went for the full 2 years. In fact, they had one guy who went 7 years with the sensor in place. Yeah. This is maybe not.
Yeah.
But there's a problem with this, and this is why this is important for you. The problem is you go to the diabetes meeting and tell them this kind of data and everything, and the doctor's saying, "Oh no, we don't like that idea at all." And this is a big surprise, because the problem is that they're saying, "Well, you mean we put one of those in a child and keep it in for a lifetime? You have to swap it out every now and then, which is no biggie. But we can't imagine taking a 10-year-old who has diabetes and putting one of these in. There's a small possibility of danger that it would erode the blood vessel wall, maybe it cause a clot, maybe something might break. Well, how often is that? That's going to happen very often. Yeah, but the times it does happen. The alternative is this child just does finger sticking. You're never going to get hurt by finger sticking. You can do that your whole lifetime, and okay, it's objectionable, but there's no really serious thing that could happen to you. And so they went to the Scandinavian heart registry, where all the pacemaker data that's collected in Scandinavia is put into this registry, and there are untoward events, they call them, that occur on the order of 1 in 10,000 patient years. We have several million patient user years now in pacemakers, right? And so the pacemaker's kind of like this. And so they have a big registry, and they have the standard rate is 1 in every 10,000 user years something happens. The doctor's got to intervene in some way, right? And so you say, well, look, explain that to the diabetes care people, and they say, well, yeah, but right now a kid has no events in their entire lifetime other than finger sticking things, right? So they're just so conservative, which, if you talk to a cardiothoracic surgeon, they'll be happy to put one of these things in place, and, they can do it with one hand tied behind their back, and then, we do this all the time. Well, you talk to the diabetes doctors, what they really do is they look at emails that say, here's my blood glucose, should I change my insulin? They don't do procedures. The diabetes doctors don't have a specific procedure they can do now, right? And invading your chest or putting one of these things in. So,
This is really an important point. and part of doing an evaluation and determining a strategic focus for what you're going to do, to know that there's a pathway for it actually to be accepted. I'll talk later about artificial heart devices. And, you know, all the interventional people in cardiology, they all deal with ischemic heart disease, but not heart failure. The heart failure doctors are not interventionalists. Here, the kidney doctors are, the pancreatic doctors, the diabetes doctors are not interventionalists. So, if you are going to, yeah, it's their culture. If you're going to introduce something, then you need to figure out a clinical pathway for intervention in what you're interested in. If there one already exists, great. If it doesn't, those folks are the ones that end up giving the papers and whatnot that introduce it to clinical use. So this is a really valuable
Important
learned lesson.
Yeah, it's a big money lesson.
Yeah.
It cost me many years of my, and it cost a lot of money for the company that made this, you know. And it's still the state-of-the-art, very best possible glucose monitoring system, you know. Blood glucose, and it's in the mixed venous blood, and
right.
Everything's great, except nobody's going to use it, right? Nobody's going to buy
it.
Yeah. Except for that minor problem.
What did they use for a power source? That sensor?
A battery.
Okay.
Lithium battery. And the idea is, okay, it's just, the telemeter is just under the skin here, so you can take this out and put it back in pretty easily, you know. It's just like putting in a pacemaker. Pacemakers are all done in the doctor's, not all, but a vast amount of them are done in the doctor's office, you know. You don't have to have general anesthesia, they just, put that lead in there and they advance it down into your heart, twist it in place, and then they make a little slit and put in the pacer unit, right? So
vena cava is right below your skin.
Yeah. Yeah. Just right, it's very accessible. Yeah. And you can't block off much of the flow by putting in this little thing in the vena cava, as opposed to other veins that are available in the body.
How large, what diameter is that in relation to the vena cava?
Oh, the vena cava is
It's large.
2 centimeters in diameter or something like that. All the blood in your body from the upper body is going through that, right? So 2 centimeters is about the right size for the vena cava diameter or something.
I'd say it's probably bigger than that.
Maybe bigger, yeah.
It's very, has a lot of compliance to it.
So this thing's just sort of sitting in the vena cava, right? And some of them get attached to a wall. They just work just fine, no problem at all.
But what Dr. Golf said about, it is producing thrombi, but the lungs are a big filter for that. And your lungs, when they're spread out, are about the same size as your tennis court. So they can filter a lot and still, you're not having an impact from that.
The important message is if you put this thing anywhere on the arterial side, it's going to thrombose, no matter what. I mean, within a matter of minutes even, Okay, well, let's use anticoagulants. No, we're not going to use anticoagulants for a lifetime, just operate this device,
Right.
And so the arterial side's out. And the arterial side, where blood goes here or to your heart or something like that. So if you make a clot, you got a big problem, on the arterial side. But on the venous side, it's all different. So that's an important message. I hope,
So
you have to understand the culture as well. Yes.
would it have been a better plan to like discuss it with, the doctors beforehand? Is that the
They
moral
were
of
so
this?
supportive of this beforehand, I
Really?
can't
Yeah.
imagine.
That's the kind of thing
We
Yeah,
discussed it like
it-
crazy.
I would agree with that 110%. That,
Yeah.
they want to be engineers.
They were just so in favor of this, they couldn't wait for it to get ready. Oh my goodness,
So do you have to prove that there's something wrong with finger sticking, that errors can occur, that mistakes can happen to prove the case for this?
I think it's a dead case at this point. Yes.
Okay, correct me if I'm wrong, but the sensor, this was an oxygen sensor or a glucose sensor?
It's got both in here, you know, I didn't show you the detail, but one in part of this is the glucose end with oxygen electrode, and then there's another oxygen reference sensor here, okay? So it's all in one little tube here.
If the vena cava is that close to the lungs, wouldn't there be an issue putting that device so close to the lungs? Wouldn't the lungs create false
Well,
readings?
your venous PO2 from the whole body is slowly going through the blood to the lungs. This is pre-lung, right?
Okay.
And it's very, very uniform. And so there was even a big discussion, why do we need this oxygen reference electrode? Because, you know, your venous PO2 at that site really doesn't change, right? So that was one part of the little story, but I said, okay, well, maybe when you exercise it will change or something, so let's leave it in there. So this is an important message for this class. I hope, you know.
Yeah.
Okay. All right, now, quickly. Oh, here's artificial pancreas, okay, with an implanted insulin pump and that sensor and what they call a P-prime controller, a control algorithm. And this shows glucose measurement in a person who's diabetic and blood sampling and sensor going along, and then there's a bunch of complicated information here. But this is insulin delivery by the implanted pump, directed by the glucose levels. And some of this has to do with basal level, okay? And then they have a proportional level, and this all has to do with the control strategy they use, which we don't need to know too much about here, but this shows the insulin delivery. And then this shows the insulins when they were sampling, just for information. There's a model that predicts how much the insulin would be there, and then they were sampling. So the point of this slide is that there have been at least one person, and probably about 20 now, who've had artificial pancreas, with implanted insulin pump, implanted central venous glucose sensor, and had their diabetes controlled. Okay, and some of these go out as far as a year and a half in about half a dozen patients. I don't think it'll ever be a product, though.
You're saying, so it'll never be approved until you can show that these patients over a long period of time have a better outcome, better health outcome, right?
Yeah,
See a lot of
how's that
money,
going to happen?
longer
Yes.
life expectancy.
Yeah. So it can be approved by the FDA, no problem, right? Everybody, except that if the docs won't buy it, and they won't tell their patients to get that, right, then that's the end of that story.
They're not going to use it until they see the benefit of the pump outweighing the risk of
Yeah.
implanting electrode.
You have to think, the way the diabetes docs think is, well, think of this 5-year-old little girl who's got severe diabetes, you know. I wouldn't do a thing to hurt her, you know. Well, what about finger sticks? Well, make a better finger stick, you know, make a smaller needle, make a, you know, because you can't kill yourself with a finger stick, right? Or you can't have any bad thing happen, basically, other than irritating lifetime of finger sticking, you know. It won't, this potential of this thing to cause something is still extremely small, but, you know,
But if you can show it extends your life expectancy by 20 years.
no deal.
Still won't.
No deal.
Even with that data, they wouldn't do it.
On this slide, is there a fixed volume of insulin in the pump, or is it coming from an external reservoir?
It's totally internal,
Okay,
and it's refilled every 3 months.
so does that mean it's an invasive surgery to refill that?
No, you use a needle, and you have to go into the clinic, and there's a nurse who knows how to do this, but they use a needle, they punch her through it's just below the surface, and there's a target there, and they punch her through that rubber target, and then they fill it in, right?
So there's pressure on it that won't let any of the insulin?
It's all designed, the pump is designed so it's not going to just squirrel all the insulin out, and it's specifically metered, and there's a lot of science in the pump and so on, but, you know. And of course, one of the problems they're worried about is, well, what if you infect the pump, right? If you accidentally get some bacteria inside that reservoir, that's a big problem, okay? But that hasn't happened in the patients they've used so far, but they're worried, oh, if this got big time, it might happen more frequently. Anyway, so, you know, medical devices, they can go pretty far. So now, I'm going to go to the, okay, so that was, I would call it at best a qualified success. And somebody would say, well, that was a big disaster. You spent all that time doing that, so. find this other set of slides that might redeem me from my big disaster here. Okay, they're going to start right here. I showed you these already. Showed you that one. That was the, okay, so here we go. So this is the alternative. Okay, this is a implanted glucose sensor telemetry system. There's a cross section of it. There's a battery in here. There's a couple of circuit boards. There's a titanium can around it, and this is a ceramic disc with arrays of sensors on it. Okay, same sensing principle as before. This is a ceramic disc with an antenna on it. Okay, so and the whole thing, you know, is about like that big around and about that thick, and this is my company across the street here has developed this, and this just happened to be from a paper we published last year, which was there. And these are just pads to anchor the thing in place when you implant it. You implant it just by making a little incision, you make a little pocket, slip this in, and you don't want it spinning around or anything like that, right? So these little pads just are there to help anchor it, or decorate it, whichever way you want to think about it. Here's the experimental subject, okay? So we have had a big pig farm going on across town with, you know, quite a few of these animals here, each one having multiple sensors implanted, right? And here's the kind of data you can get from that type of system. Okay, I won't show you too many technical slides, but this is an example. This is days on these axes, and it's just one big continuous strip after another here. This particular segment starts on day 167 and goes all the way down to day 195 in planning time. The red lines are the transmitted signal, glucose sensor signal. And the blue dots are blood glucose signal, blood glucose drawn discretely from the animal to compare. And so the story is this: a pig, pigs of course eat vast amounts of food, but it doesn't cause their glucose to change. They're different than us, okay? You saw those big spikes that people have? Pigs never do that, right? They eat like crazy, but they are highly efficient at converting food directly into body mass, right? With no excursions, unnecessary blood excursions needed. So in that sense, pig's not a great animal for testing this. So you've got to give a big glucose injection directly in the bloodstream to cause their blood glucose to move. Pigs are highly efficient farm animals. And you can't get their glucose to move. So they're baseline all the time, basically. Except here's where we just, you know, give them a squirt. And this shows when we needed to calibrate this thing. This shows, okay, there's a little change going on here. We recalibrated it on day 186. Okay. And this doesn't require daily finger sticking to operate this kind of sensor, I have to emphasize. So this is just a segment of this, and then we kept this animal going as an example for a year. And then we made him diabetic by giving him an injection of streptozotocin. This is a very specific molecule that goes to your beta cell, wipes out the beta cell, leaves all the rest of your pancreas totally functional, intact, everything very happy, except that suddenly you're diabetic. Okay. So this animal was converted to diabetic at day 360. And then here's the same kind of story. These are panels that are all connected together, and this one starts on day 373 in that same animal. And yeah, this goes to, it goes to day 376. And they're spread out a little bit more, but the scale is the same as the previous one. So now you can see that nice flat line in the pig. You can see what kind of glucoses are going on. Each one of these is one full day panel, just blown up to show the details. So this guy's glucose is going okay for a while, then it goes up spontaneously and comes back down. What's not shown in these pictures is the activity level of the investigator. Okay? If the blood glucose starts going down in the middle of the night, somebody's got to run in there and give this guy some glucose. Okay? Because he could die before morning. Okay, and the company guys do this, right? That's what they're paid for. The guy's gonna die overnight because of hyperglycemia, so you gotta run in there and give him some insulin. And so 6 months of that lifestyle for the investigator is about enough, you know. So, so that's one of the problems, right? Is how do you keep going with these kind of experiments? Yeah, so we don't ask students to do these studies, obviously. I mean, you know, so they have to be somebody's paid. They're going to be there all night long watching and all that stuff. And here's another guy. This one. Instead of the first 1 year of non-diabetic phase, we went out like 10 days or something like that, and then we made him diabetic. And here's what this guy's example is, right? You can see it maxed out 400 here a number of times, and it hit the bottom here as well, and it's cut off electronically at 50. So there's a big difference between the non-diabetic and the diabetic peak, okay? This is one of the messages here. And and the other message is an implant in the tissues can be there for a long period of time and be totally functional and effective. And most people say, well, how's that possible? Well, this gadget has the reference oxygen sensor and the oxygen-based glucose sensor on the same implant. And of course, yeah, there's a capsule that forms around this, no doubt, right? But that capsule affects both sensors simultaneously, right? So it does reduce the oxygen transport to both of them. And, you know, you take the difference and you can largely eliminate the effect of the capsule. So it's kind of a design strategy. And this is proof that it works okay. I won't bother with it. So I made my point here, I guess. Now, these are the people that helped do this, and here's a picture of somebody in this room. Very important person in this whole thing. So I just have to say that the students have been fabulously helpful, and there's some people of glycans in there also. Now, I'm going to stop again and take a little break, and I want to tell you there's a couple of business issues that I want to make sure are really. How do you get your company to survive as long that you can make one of these things? You've got to explain that because that's really important. We're not going to talk about sensors per se or diabetes anymore, and I'm going to talk about how did the company last long enough to make one of these things. That's the next part of this. So let's take a little break and we'll resume in 10 minutes and then we'll be where we want to be on this.
Yeah, but that's 7th class of that picture.
Oh, those are those.
Aviators.
You look just exactly the same.
Huh?
This is a
Wow.
long time ago.
This is a long time ago.
Computer hasn't aged in the slightest, you know.
Dr. Sobin still there.
Sid Sullivan, yeah,
Yeah, wow.
passed away some years ago, but actually 10 years ago now, right? Yeah. And,
Joe Baker's name?
Joe Baker. This guy got a brain tumor in the meantime, right? He's still alive, but he had this giant tumor and it stopped working. This guy's Professor Irvine, Glycens employees, you know, great people. This guy's still lab manager lady, and she's with me still. Anyway, I was, you team of really dedicated and excellent people to pull this off. And I want to give credit to these people for all this stuff. So let's take a little break, and I'll make it quick and finish up. And John might have a few things to say.